Hi everyone, welcome. Welcome to Shoals Marine Lab's weekly marine science seminar. I'm Jennifer Seavey. I'm the Executive Director of Shoals. For those of you who don't know, Shoals is the largest and oldest undergraduate focused marine lab in the country. It's jointly operated by UNH and Cornell, and we're located on Appledore Island, which is 10 miles offshore of Portsmouth, New Hampshire. Every summer or every typical summer, I should say, on the island, we host these talks that we call Rock Talks for the rocky shore on Appledore. We host them for our entire island community, but this year we have brought things off the island and online, and we're really excited to have you with us tonight to share this excellent talk with old Shoals alum, faculty and researcher. He was last out, we decided, in 2014, so we're glad to have him here with us tonight. So our format for tonight's talk is 45 minutes of lecture, followed by 15 minutes of Q&A. If you have a question, which we would love to hear your questions, please use the Q&A box that's at the bottom of your screen, okay? Click on that, enter your question, type it in. Don't raise your hand, just use the Q&A box, and that's how we'll get to your questions. I'll read them out loud to Ian and he'll answer them. If you have any technical problems at all during the talk, just also use that Q&A and we'll help you out with that. So tonight's speaker is Dr. Ian Hewson. He's an associate professor at the Department of Microbiology at Cornell. He also serves as a director of undergraduate studies for marine biology. His research focuses on marine diseases and their relationship with environmental variation and the relationship between disease processes and microbial ecology. Ian has an undergraduate degree from the University of Queensland, which you will soon detect in his accent. And he has a Ph.D. from the University of Southern California. And he did a postdoc at the University of California at Santa Cruz. So he is really well known for his work on sea star wasting disease, which I'm very excited that he's going to talk to us tonight. And his talk is titled Red Herrings, Misleading Results and Redefining a Disease, Sea Star Wasting in a Changing Ocean. Thank you so much, Ian, for doing this talk, and I will turn the floor over to you.
That's great. Thank you so much for that introduction. It is a great honor to be here with Shoals in this strange new world in which we exist. It's great to see so many people able to tune in. Tonight, what I'm going to do is to describe some of the research that I've been involved in for the last 7 years since sea star mass mortality happened on the west coast of North America in 2013. It's been an ongoing process. It's been a big evolution of our understanding about diseases in general, and I hope to communicate that with you today by this presentation. So I'll just bring up my actual presentation here, and let's see, just give me a second here. So sea star wasting is a condition which has affected a great number of sea stars, starfish, if you want to call them that. We don't usually call them fish because they're not fish. Asteroids is another word, although a lot of astrobiologists get a little confused when we talk about asteroid-associated bacteria. As I was saying, you know, this is a disease which we had, it came upon us very suddenly, or so we thought, in 2013, and it's been a big job to figure out what might be causing it. And the take-homes that I'd like you to learn from this lecture today, or this seminar, is apparent disease, even when it's highly transmissible, or it looks to be highly transmissible, might actually not be pathogenic, and that certainly seems to be the case with starfish wasting. Temporal changes in microbial communities and genomics, looking at their DNA sequence, can provide interesting insights into disease processes. And finally, many biological phenomena in the ocean co-vary with microscopic physicochemical change. And in other words, the scale at which you're looking at a disease sometimes is on the microscopic level as opposed to the whole animal, the whole ecosystem. The processes that happen on the microscopic level are what influences the health of animals in the ocean. So the first, I've said, it's a disease. You might have heard of sea star wasting referred to as a condition, as a syndrome, as a disorder. Oftentimes these terms are used interchangeably. but many imply that it's some sort of infectious or transmissible agent, all right? When we say disease, a lot of people think instantly that it's some sort of pathogen, that it's a microbial pathogen that's causing it, and it can easily transmit from an infected to an uninfected individual, kind of like what COVID-19 or the 2019 novel coronavirus does. And it's a little bit broader than that. So diseases are defined as any change in the normal function or structure or any part, organ, or system of a living body. So that's not entirely sort of consistent with it being a pathogen in all cases. This is actually a definition that was put out by the Wildlife Society. It's a big sort of disease ecology group. And it's interesting that they sort of follow up with this initial description as it is also defined as a pathological condition occurring in the susceptible population. So in other words, a pathogen-derived thing. And then it says pathogens are natural components of ecosystems that may be limited by environmental conditions and so on and so forth. It's just really interesting that the focus on disease tends to revolve around pathogens, and perhaps not so much on those diseases which are generated from the animals themselves or by the environmental conditions in which they live. And today I'm going to give you an example of a disease that looked a lot like a pathogen-driven disease, which in fact we now believe is not. So why are we interested in echinoderms? Why are echinoderms so important in the ecosystem? Well, first off, they're economically important. All right, there is a very active fishery of urchins. There's about 110,000 tons of them, which are harvested every year. Holothurians, there's about 20,000 tons. They're also ecologically important. So certain types of echinoderms, notably asteroids, are keystone species. But also holothurians and echinoids are very important as consumers of detritus as well as in terms of herbivory. And what's really interesting about echinoderms is that they experience these mass mortality events. They're known as a boom-bust phyla, all right? So that what we mean by that is that they undergo these very large population explosions, which then subsequently collapse and they go away. The reason for that is often sort of a limitation imposed by the environment and the amount of food that they have, but also there's sort of disease events like what we're investigating here that are poorly resolved. And also that echinoderms are very sensitive to environmental perturbation. So in other words, you can have a very small decrease in water quality, and that can lead to a huge increase in mortality in echinoderms. So they're fascinating animals from economic as well as ecological standpoints. We've had several key questions about sea star wasting since we first observed it in 2013. And this is how I'm going to structure this talk. The first is, what is sea star wasting? And I put in brackets there disease, just to remind me that we don't actually know that it is a pathogenic disease, okay? It's just a condition which is abnormal in these animals. The second is, what causes sea star wasting? That's kind of the $64 million question. What is the cause of it? Is there anything we can do about it? All right, and I'll give you some evidence from multiple different studies that suggest that we now know what might be causing it. And finally, how long has sea star wasting been around? All right, I mentioned that 2013, there was a big outbreak of it. That's the first time we noticed mass mortality as a consequence of sea star wasting. But has it been around before that? We have evidence that, in fact, it has been around for a really, really long time. And then finally, I'll give you some insights into what's next in terms of our investigation. So sea star wasting sort of appeared out of nowhere, suddenly in summer of 2013. This is a map of the west coast of North America. Up the top there, you can actually see Alaska. Down in, you can see central California and the Pacific Northwest here. This is known as the Salish Sea. Sea star wasting was first observed on the Olympic coast of Washington State in June of 2013. It then sort of spread around the Salish Sea, seemingly spreading from site to site. It also seemed to occur at the same time in central California, and then it spread to Southern California. It sort of bypassed this big, long area of coastline in Oregon. Lots of people monitoring the intertidal there, so it's not as though people weren't looking. And then eventually it made its way there, and eventually it spread up into Southeast Alaska, and it was found, the furthest point is out on one of these islands in Cold Bay in Alaska, I believe, or maybe that's the Alaska Peninsula, sorry. So it's basically sort of the widest marine disease event that's ever been documented, all right? This is by far the biggest kind of event, the most intensive as well, the most individuals that have died within a species of naturally occurring marine invertebrate. But what's really interesting is that it was an asynchronous appearance along the coast, all right? So I mentioned that it sort of skipped parts of the coast and then came later. If we were looking at a pathogenic disease, we'd expect it to move from place to place along a coastline as it spreads from infected to uninfected individual. This didn't really follow that. It seems to sort of move from one point and then leapfrog entire stretches of coastline. And also what's interesting is that when it first came about in summer of 2013, there was no apparent correspondence with temperature anomalies or El Nino years, all right? So it wasn't as though there was a very large, thermal mass of water that was very warm that was moving on shore that was causing it. That actually happened about 1.5 to 2 years after the onset of this wasting condition. And so it was a little bit unclear as to what was actually driving it. It is a very drastic disease for the animals. At the top here, you can see a photograph of the sunflower star Pycnopodia helianthoides. What happens is they go from completely healthy or asymptomatic. They lose their turgor. They basically deflate. Their arms curl and twist. That's a very subjective disease sign that hasn't really been firmly nailed down. And then they have this process whereby their body wall tissues discolor. They have these lesions that form. The arms drop off and crawl away. That happens in some cases. And then eventually the body wall disintegrates, the surface sloughed off, the organs protrude, and essentially the animal decomposes. So here you can see a wasting star in the middle, all right? And at the bottom here, you can see a pile of ossicles and bacteria that are eating the carcass of the animal. It's something called vegetator, basically a sulfur oxidizing bacteria. And the process from going from completely healthy to completely dead and gone is about 2 to 7 days for these animals, all right? So it's a really, really rapid thing. It caused huge numbers of these animals to disappear all at the same time. It also affected a very large number of species of sea stars. So 20 species in total had some sort of wasting sign associated with them. What was interesting from the early investigations is that larger individuals and more active or predatory species were the ones that were affected first. So at the top here, you've got Pycnopodia helianthoides. Underneath here, you've got, to the right of that, you've got Pisaster acraceus, the ochre star, Pisaster brevispinus. That's Patrick, the starfish from SpongeBob, just in case you're wondering. And this is Evasterius. All of these ones that were very active and very large species were the ones that were disappearing very quickly, whereas those which were relatively small and those which were relatively slow moving were the ones that were not dying off as fast. It's also interesting that this condition affected a very disparate phylogeny, right? So while some of these starfish look the same, they have more or less the same body plan, they're actually very different from an evolutionary standpoint, all right? So this one here, Solaster, is actually distantly related to this one here, all right, Pisaster brevispinus. This one, which looks very different from the rest, sunflower star, is actually really closely related to the ochre star to its right. So it crossed across these big taxonomic boundaries of different organisms. And it has, unfortunately, led to the functional extirpation of several different species, but sunflower stars, the Pycnopodia helianthoides, at some sites in the Salish Sea are completely gone. We can't find them anymore. And in terms of the overall distribution, the overall loss of starfish across the entire Pacific coast, it's about 10 to 90%, depending on where you are, of the total starfish that were present are gone, all right? So millions and millions of starfish have basically died as a consequence of this, or they've disappeared. So what is wasting exactly? Well, the first thing is to realize is that there's no defining characteristics of wasting. There are no characteristics that you can look at from a microscopic level, from a tissue level, and say that that is a wasting starfish, all right? So there's been quite a few studies using a tool called histopathology, and those are images that you can see to the right of this, that basically look to see what was different about sort of the surface of the animal, the tissues that are basically on the skin. And all they could find was that, and it comes up with some very consistent findings here. Selenocyte aggregation, immune cell aggregation, edema, swelling, all right, cleft formation, so basically their outer skin stripped away from their underlying sort of bony structure. You think about it like a bony structure. It's actually a whole mass of little ossicles that are sort of embedded with each other, and some sort of necrosis of their epidermis. But there are no disease signs that are characteristic of this disease. In other words, there's no pathonomic signs of it. And there's also no case definition for any species affected, right? A case definition is basically a single species with the disease that's being described from a tissue level to say that is definitely that disease, okay? But what we can say is a very common characteristic are these lesions. And the big question is, what are the lesions? So when you look at a lesion, and this is an up close of Pisaster acraceus, this is actually a lesion here. What you can see is that it's simply the loss of this epidermal tissue. That's this purple stuff here. And what you're looking at in that white lesion is actually the underlying body wall, which I was just saying is actually, it's not really a skeleton. It's actually sort of a mass of little ossicles that are kind of intertwined. And so we sort of looked at that and we thought, well, something's obviously causing the tissue to slough off. And we wanted to know exactly what it was. Another important thing to realize about starfish is that when we looked at those lesion margins and we looked at wasting starfish and healthy starfish and compared them, we compared the genes which are being expressed by both. What we can say is that basically the wasting stars are destroying themselves. There's a process called apoptosis or programmed cell death, whereby basically cells are triggered to basically implode and the internal components are recycled. We see a lot of genes involved in structural rearrangement, autocatalytic tissue degradation, and loss of energy metabolism in these animals compared to healthy animals. And so it basically is consistent between what we're seeing by histopathology, tissue degradation, and microscopy is reflected in gene transcription. So what's causing it, all right? That's the big question. We know that the animals are sick. We know that their tissues are sloughing off. What is actually causing it? And like I said, that is the big question. So what causes sea star wasting? So it was for a long time, and it still is in certain realms. Basically, it's thought to be a transmissible disease. Individuals collected and placed in captivity would die very rapidly, especially under warmer conditions. A big piece of evidence that we thought it was transmissible came from observations at the Vancouver, Seattle, and Monterey Bay aquariums, which found that the animals in their captive collections they actually died at the same time as the animals on their intake pipes. So basically these big aquaria, they suck in water from outside, they circulate it around their tanks and they pump it out. When the starfish were dying on the outside of those intake pipes, the starfish in the aquarium were dying. So it was sort of thought that it was actually something that was making it through those pipes, perhaps their filtration system, and infecting their captive populations. Another interesting observation was that ultraviolet light treatment of that water that they were bringing in improved the survival and basically didn't cause disease. And so if you hit it with UV light on the in current, it would not lead to disease in the captive populations. And interestingly, the one place that has a very large aquarium in California, which is the California Science Center in Los Angeles, they saw no mortality at all right? And they use artificial seawater. So it was sort of, if you piece that together, it was pretty clear that there was something which was perhaps coming in from outside the aquarium into the aquarium that was killing everything. And so back in 2013, my lab actually went through and looked at different types of viruses, different types of bacteria, and different types of eukaryotic microorganisms. So those are protozoa and fungi. And we figured out that there was a C-star, there was a virus basically associated with the wasting tissue. This is known as the C-star associated densovirus, SSADV. We concluded that wasting was associated with it. We basically pieced it together. That's its map of its genome there. So it's a really, really small virus. To put this in perspective, coronavirus is about 4 or 5 times this size. This is really, really tiny. And we sort of also performed an experiment whereby we took some wasted star tissue. We basically mashed it up. We got all the viruses out of it, or so we thought, and we put it into a healthy animal, and they became sick, all right? And the sort of sickness that it caused were arm curling and tissue loss in about 80% of them, accompanied by the elevated loads of this one particular virus. So we had at the time of 2014, we published this in Proceedings of the National Academy of Sciences, a good idea that there was probably a virus involved, and it was probably, this is the best candidate pathogen for it. However, further research that we did basically did not back that hypothesis. So between 2014 and 2018, we performed another 8 different experiments of exactly the same type, and we could not basically elicit wasting in any of them at all. The other sort of red flag that we had was that many asymptomatic stars had this SSADV, and many of the wasted stars actually lacked this virus altogether. And so we sort of took a step back and we looked again at sort of the primers. We'll be using something called quantitative PCR to look at viral load and prevalence. And we sort of looked at sort of more the diversity of densoviruses than the ambient population. And we found that it basically didn't hold up, all right? There is no association between SSADV or any densovirus for that matter and wasting as far as we can tell. So brought us back to the question, you know, what is actually causing it again? And we published this in Frontiers in Marine Science in 2018. There was a series of reports that came out from about 2015 onwards, which were talking about environmental impacts. Obviously, climate change is influencing the oceans quite heavily. Elevated temperatures in the oceans, as well as the temperature-related effects, were thought to be responsible for it, perhaps. There's a few studies that came out. For example, higher temperatures resulted in faster wasting and death in aquarium experiments. Higher temperature was accompanied by wasting and field surveys in San Juan Islands. That was done by Drew Harvell's lab at Cornell. And in the field, lower temperature and lower pH, right? So that's in complete contrast to those first two studies, were accompanied by wasting in coastal Oregon. Remember, coastal Oregon was that population that basically wasted almost a full year after the ones in the Pacific Northwest, or sorry, in Salish Sea. And we actually looked at sort of other physicochemical and meteorological conditions which might be accompanying this disease. And we found that wasting onset was preceded at about half of the locations that we surveyed by below 5-year average temperature, all right? And so what this means is that there's no universal temperature wasting relationship, all right? So that had our scratching our heads, basically. So we sort of took a look at some of the data on temperature in 2013 at a certain site in the Salish Sea, as well as the onset of wasting. And we came up with this hypothesis that rapid temperature shifts. So we did see some temperature go up really fast and then really far down and then really far up again. And we thought that perhaps that was the cause. They just couldn't deal with the stresses of this constantly changing temperature. And so what we did is in 2018, we went out to Santa Cruz and we collected 24 Pisaster acraceus, the ochre star. You can see them there in sea tables. And we noted no wasting in the field, populations at the time of collection. But what was interesting is that after 14 days, so what actually happened was we put them into sea tables, we came back to Ithaca, we were expecting them to acclimate for 2 weeks, and then we're gonna go back. So guess what happened? While we were in Ithaca, the animals wasted away, all right? In totally unremarkable flow through sea tables, not a lot of external stimuli. So it seemed that there was basically something else that was at play, perhaps the stress of collection. So we redesigned the experiment. We went back as planned, and we performed 4 experiments to examine the impact of various different stressors. Desiccation, was it the fact that they were sort of exposed to warmer air outside for a longer period because of weird sort of temperature anomalies on the West Coast? That might have caused it. And you can see here on the right-hand side, this is how we did it. We actually took the starfish and we stuck them on these plastic plates and left them out in the sun for an hour and then put them back in aquarium. Aquarium water replenishment rate, we wanted to see whether the flow rates of aquariums were actually influencing wasting onset and severity. We also had this sort of idea perhaps that by putting starfish into sort of a small container, they might cause some sort of abrasion and damage, and that might lead to wasting. So we actually physically abraded several of the starfish and compared them to some that we hadn't, or just abraded them with seawater. And then we also performed another challenge experiment like what we did in 2014, except this time what we did, so the control for the 2014 experiment was a heat-treated tissue homogenate. In this case, we actually treated the homogenate with proteinase K, which selectively gets rid of proteins. And so we were seeing whether proteins from wasted starfish were capable of inducing wasting in asymptomatic animals. And the results of it were that pretty much everything causes wasting, all right? We found a significant difference between the rate at which the starfish got sick in low flow rates compared to high flow rates. So in high flow rates, they seemed to be okay. Low flow rates, they were not. Also, when we blasted them with seawater, they survived longer than when they were blasted with seawater in this sort of pedicelaria that were taken off of other starfish. Also, desiccation seemed to influence it as well. And notably, it seemed as though the tissue homogenate, whatever proteins were in there seemed to be causing faster wasting, although really not that significant. But the largest effect on wasting speed was a consequence of low replenishment rate of seawater in aquaria. So we were interested in the temperature sort of hypothesis and also to see what actually happened in nature. Like, what is a typical environment of a starfish as they occur in ambient conditions? And so we deployed a couple sensors. These are HOBO sensors, which measure temperature and salinity. We placed them in the intertidal zone around starfish populations, a place called Davenport in California. And what we found is this. So here you'll see that the aquariums are in the green line as well as the yellow line. They don't change much in the aquarium system, all right? Whereas if you go to the field, they undergo these huge shifts, absolutely huge, all right? Right up to 33 degrees Celsius at a low tide in the middle of the day, right down to about 7 degrees when they're immersed at night, okay? So if we were thinking that perhaps it was a temperature change thing, this basically indicates that they are capable of dealing quite well with huge temperature differences, and so it's probably not that case. Interestingly, the desiccation experiment that we ran, the starfish reached about 34 degrees Celsius in air. So it's really, really hard to reconcile temperature as the sole cause of wasting, given that they're used to being out there in really rapidly changing conditions. So that's sort of led us to sort of look again at the microbiology involved in sea star wasting. Back in 2014, we were really restricted to comparing symptomatic and asymptomatic animals, all right? Most of the collections, because the wasting disease had already taken off, were of animals that were dead or close to dying versus those who were totally healthy, okay? And one thing we didn't really have a really good handle on is how things change from a completely healthy animal to a completely wasting animal, which is absolutely critical as it turns out. So many stressors result in sea star wasting in the aquarium, like I mentioned, desiccation stress, I spelled that wrong, I know, abrasion, proteinaceous homogenate from a wasting star, elevated water temperature, the homogenates of wasting star. And so, you know, it wasn't really clear what was going on. So we thought, why don't we just look at, you know, wasting progression, the microbial ecology as things go in the absence of external stimuli from completely healthy to completely diseased. And here you can see a little schematic that I've drawn here, where basically we have healthy tissue, we've got their normal microbiome, we've also got mucus, which is being released from it, supporting the growth of bacteria near to the animal surface. Viruses exist there too. And under lesion margins, we would expect to see the pathogen. And then in the actual lesion itself, we would expect to see almost nothing because there's not a lot for them to grow on. So what we did is we took animals, we took a total of 6 of them from Davenport. We were interested to see what actually occurred in damaged tissue as opposed to a lesion margin. And so what we did is we made a little artificial scar. We nicked the surface of it with a small syringe, a small needle. And then we monitored for the appearance of natural lesions away from the scars. And we were taking tissues from controls, tissues on the same animal away from the scar or away from the lesion, as well as from the actual lesion margin, as well as from the artificial scar. So it gives us a good way of sort of comparing what might be growing naturally on the animal, how it changes as the lesions sort of start, and how that's different from it just being damaged exterior, all right? And the first thing we did was to look at viruses, okay? And this is using an approach called metagenomics. And this entails taking all of the viruses out of a tissue, grinding them up, sequencing their DNA, and then trying to work out who was there to begin with, all right? And this is a little bit of a complicated figure, but what I'll tell you is that in this heat, it's called a heat map. Each one of these red squares indicates when there's a lot of something, all right? And if it's black, it means it's not present, and anything in between is sort of a gradient. Each one of these rows on this figure indicates a different virus. And then I've grouped these together with these non-wasting stars. So on the left, these animals were totally fine for the duration of our study, whereas wasting stars were the ones that became sick. And we took several samples. So you have the time 0, this is the initial sample. We've got the control at the first lesion appearance. So that's tissue not on the lesion, but away from the lesion on the animal when it first formed the lesion. We've got the artificial scar, we've got the natural lesion, those are those ends at the end there. And a couple of things pop out, all right? There were a few, a handful of viral contigs, we call them because they're just contigs, but they're actually viruses that increase in abundance over time in wasting lesions, all right? So here you can see all of these, which are indicated by this last column here, NTF. They were not there at the beginning, and they certainly seem to increase over time. However, if we look at the non-wasting stars, they also appear to be there, and they just seem to increase over time, even in asymptomatic tissues. And there's no consistency between individuals. We don't find the same virus occurring in 2 different starfish in association with wasting. And what's really interesting when we looked at this, the number of viruses which became really abundant as animals died just skyrocketed, all right? So a lot of viruses suddenly appeared in the tissues, and it basically suggested that there was something else going on during the disease process which might support the proliferation of viruses, all right? So no evidence for any viral contig associated consistently with wasting, but wasting may generally stimulate viral proliferation, all right? And keep that in mind because we're gonna come back to that a little bit later. So then our focus turned to the microbiome, and here we're talking about bacteria, all right? And what we did is we took samples of the epidermis and body wall. This is from that first experiment where the animals died when we weren't there. Fortunately, we had an awesome graduate student at the time, Lauren Schiebelhut, who went down to Santa Cruz from UC Merced and sampled them as the animals died. And what we looked at, we found that there were sort of bacterial communities changed from T0. So these are these ones in I guess that's beige, through to the formation of lesions, that's the light blue, and then through to their final time point when they died, and that's the green. And we looked at what types of bacteria increased. So we had a couple of well-known weedy species. So Vibrio is one that definitely increased over time. Tenacibaculum is a type of Bacteroidetes, which is often associated with nutrient-rich environments, and it's no surprise that in a decaying animal it was increasing as well. And then we had this one, Clostridia, all right? And Clostridia increased. Clostridia are very interesting because they have some interesting features about their biology that we hadn't really sort of seen before. I'll just mention also we looked at samples from epidermis only, and in this case we were looking at control and lesion. They tend to form different types of bacterial communities. And again, we saw this sort of increase in Tenacibaculum in those. We also saw this increase in something called Pyrrhella, but we saw this increase in D-sulfovibrio. Why are they there? All right, D-sulfovibrio and Clostridia are both strictly anaerobic bacteria. Why would we find anaerobic bacteria in an otherwise well-flushed, very, oxygen-rich environment? And in general, we saw this sort of progression from strictly aerobic taxa to facultative anaerobes and microaerophiles to strict anaerobes. So why would strict and facultative anaerobes thrive on starfish? You know, seawater is mostly saturated with oxygen, right? It's usually about 10 milligrams per mil. So how can strict anaerobes actually survive? And so it took a lot of sort of head scratching, but the answer is probably boundary layers, all right? So what I mean by boundary layers is that these starfish have sort of abundant terrain, all right? They're not just a flat surface for the most part. There's actually pits and valleys. And within those pits and valleys, there's a lot of material that bacteria consume. The starfish is also respiring and using up oxygen. And so there's real potential for there to be depleted little zones of low oxygen conditions. And so basically we focused our attention on that. We started to think perhaps this might be what was relating to wasting disease. So our next sort of experiment that we ran is how does low oxygen actually influence wasting, all right? And what we did is we took some Asterias forbesii from Bar Harbor up the road from Shoals, and essentially we put them into aquarium systems. We separated them out into little Tupperware that had a whole bunch of holes punched in it. And we subjected about, it was 12 of them to hypoxic conditions, roughly about half the normal concentration of oxygen. And we had the rest of them as controls, just in a well-flushed, well-circulated aquarium system. And what we found is that lesions similar to wasting appeared rapidly after 3 days at about half the normal oxygen concentration. All right, so here's a survival curve. You can see this is the proportion without lesions, so this is the total number that are healthy. The control ones stayed healthy the entire time. The ones that were subject to this half the normal oxygen conditions, they died really rapidly, all right? And what's interesting is that it was also accompanied by a 1 to 2 order of magnitude increase in bacterial abundance on their surface, all right? And so that sort of gave us some evidence that perhaps bacteria might be driving this phenomenon, perhaps by, increasing their abundance, using up oxygen on their surface, and then basically causing wasting processes. So it turns out that oxygen is one of the most difficult things in oceanography to measure, particularly on a really fine resolution. One thing you can do to try and work out what this boundary layer looks like is to use something called a microelectrode, all right? And this is this funny little sort of pipette-looking thing. And essentially there's a platinum wire that runs through it, and you use this microscope stand and a little fine scale knobs to sort of move it down onto the surface of the animal and then move it back. And it gives you a little sort of profile of oxygen. The problem with this, as we learned, is that these are really expensive. They're about $1,200 US each, very, very fragile. They break super easily. And starfish have this tendency to know when there's a probe around and decide to move their arms and break the tip of it, making it unusable forevermore. All right, so we learned very quickly that this is a very expensive branch of science. But what we did learn from it, in addition to the expenditure, is that oxygen is depleted tens to hundreds of microns above the epidermis. So this is basically the depth. So here you have the Pisaster surface indicated in purple here. Each one of these lines represents a different oxygen profile. So 2 different animals, you can see that under normal conditions, it's relatively, on seawater, it's relatively linear. It's sort of linear up and down. But you can see that there's some depletion the closer you move to the animals. This becomes worse over time. And in fact, when you add glucose to it, which is usable by most marine bacteria, about 40% of them, it results in this very rapid depletion of oxygen near their surface, all right? So this actually stimulated our thought process that perhaps asteroid surface hypoxia could be causing wasting disease. So I'll just step back a little bit and talk a little bit about bacteria. Bacteria are hugely abundant in the ocean. Every drop of seawater has about 1 million bacteria in them. Almost all of them are non-pathogenic, all right? They're just there to chew up what's known as dissolved organic carbon, all right? And dissolved organic carbon comes from various different sources, all right? Almost all of it in marine habitats, particularly those offshore, comes from autochthonous sources. So that's phytoplankton that are basically spitting out carbohydrates and all that stuff that they photosynthesize. They release it immediately and it's taken up by bacteria. Some part of it comes from allochthonous sources. So that's terrestrial runoff and other terrestrial sources, geological weathering. So basically the sources depend, the ability of bacteria to use this depends entirely on where it's from. So we tend to find that coming out of phytoplankton is really usable by marine bacteria, whereas that coming off of basically terrestrial environments is not as much. And the uptake generally follows a sort of path from copiotrophs or weedy types of organisms that respond really fast to high organic matter conditions through to oligotrophs or things that are relatively slow growing. The taxa that we see by 16S on the surface of starfish are in fact copiotrophs. So what stimulates bacterial respiration? It's simply the addition of organic carbon, right? So bacteria plankton rapidly assimilate and respire DOC. In fact, in this aquarium system, we accidentally switched off the pump, which is keeping it aerated, and we added a whole bunch of peptone to it and reduced by 37% after only 25 minutes and 200 liters. So that's a huge depletion of oxygen. So what we think is that chronic loading of dissolved organic carbon may lead to persistent hypoxia near animal surfaces. So we then went about and we tested whether DOC actually does cause wasting. So what we did is we took variable sources of organic carbon, including peptone, which is a peptic digest of casein. We also took Dinelliella, which is a type of phytoplankton. We took that culture and used that as a carbon source. We also took some coastal phytoplankton and we added it to various starfish in aquariums. And we found that all three of those organic matter amendments caused this sort of more rapid wasting compared to controls. And we also saw this sort of increase in bacteria on their surface, this very rapid increase within the first 48 hours, and then a very rapid decrease after that. And it turns out that bacteria that are anaerobic have very slow growth rates. So they maintain lower cell abundances, but the copiotrophs die off because of those low oxygen conditions. So why are sea stars so susceptible to hypoxia? Unlike other types of invertebrates that have gills or gill-like structures, sea stars respire through their skin. They don't actively ventilate, all right? Gas diffusion occurs across their outer epidermis, and it has to make it all the way into the middle of the animals in order for it to satisfy respiratory demand. And what's interesting is that we measured oxygen demand for respiration, which is at or near the possible diffusion rate of oxygen across the epidermis. So here we have the individual mass of the animals, the respiration weight. Bigger animals tend to respire more per unit of their weight. And so if you have a lower oxygen, which is depleted at the very surface, the maximum diffusion rate is significantly decreased, and they can't meet oxygen demand. In fact, for some species, and note these are Asterias forbesii that occurs on the East Coast, as well as Physaster cracius, it's actually they require a lot more oxygen than they're actually capable of diffusing. We think that that relates mostly to the sort of presence of papillae or other little structures that they can actively pump water around with their cilia. And some of the species where basically they don't have much of an oxygen demand. Interestingly, these two species at the end here were the ones that were not as badly affected, if at all, by wasting disease. All right, so we sort of had this idea, okay, so we got some sort of depletion of oxygen at their surface. Why did we see some species more heavily affected than others? Some species were not affected that much at all. And so we sort of had this idea that perhaps we started to notice that the animals that were less affected were the ones that were relatively smooth compared to those which were, relatively rough. Those were the ones that were more heavily affected. And so it comes to this concept of rugosity, all right? Rugosity simply means the corrugatedness of a surface. It's a physical property of them. And wasting affected species have complex surface properties. They're more rugose compared to those which are less affected. So Dermasterias is one that's taken over on the West Coast now, relatively smooth animal. Physaster brevis spinus, heavily affected by wasting, very fluffy, lots of topography there. And the presence of the structures, be they paxilli or pedicellariae, these little claw-like things, impacts the boundary layer distance. So here's a figure that came from 2017 from a paper. Seagrass beds, all right, you have this sort of boundary layer on top of the seagrass, which depends on the length of them as well as the shoot density. All right, same thing happens on the surface of a starfish. But we wanted to measure that empirically. We couldn't just hand wave about it. So how do you compare wasting susceptibility to overall rugosity? The answer is you have to work out their total surface area and their total rugosity using imaging tools. And in this case, we use computed tomography, all right? So this is the same CT scanner that you go through when you're not feeling well, or you take your pet to. This is actually at the Cornell University College of Veterinary Medicine. We collected a whole bunch of different types of sea stars. We fixed them, and then we put them through the CT scanner. It resulted in a lot of 3D images like this, from which we could calculate the total surface area, and we could also calculate the relative rugosity between species. And we also did something called microscopic computed tomography. So you wanted to see at the really, really fine scale. The previous CT scanner that I showed you only goes down to 400 microns. This actually goes down to 20 nanometers. The previous one was 400 nanometers. And we found indeed that wasting affected stars were more rugose than those which were not affected, all right? So what this is telling us is that there seems to be some relationship between the physical structure of a starfish and its relative susceptibility to disease. All right, so we have this evidence. We know that, okay, so we know that in contemporary starfish that are wasting today, there seems to be some sort of oxygen depletion that's probably caused by organic matter enrichment that probably leads to bacterial explosions on their surface, leads to lower oxygen conditions, and that causes wasting. But the big question is, do we see evidence of this effect in 2013? And it's taken a lot of data mining to go back through lots of data sets to figure out that in fact we do, okay? So this is actually a paper which is looking at metatranscriptomics that I published with Brent Goodenkauf in 2014. And what you can see is that the abundance of strictly aerobic taxa was really, really high in average in healthy individuals, but it was almost absent in several of them and very much lower in the diseased individuals. Whereas facultative anaerobes had the opposite effect, all right? We found this proliferation of anaerobic taxa, and also we can see that in some of these other types of bacteria for which we have some quantitative data. We tended to find that wasting asteroids had this proliferation of copiotrophic bacteria, and in fact, they could have led to the abundance of anaerobic bacteria, facultatively anaerobic bacteria. We also have more evidence for hypoxia, all right? Larger stars died faster than smaller stars. There are some caveats associated with that, depending on detectability. So smaller starfish are harder to see in the intertidal, but certainly it seems to affect the bigger starfish more than the less one. And that relates to their surface area to volume ratio, okay? So basically, larger animals have a very much lower surface area to volume ratio. And so they basically are unable or less efficiently diffuse oxygen into their bodies compared to those animals which are really, really small. The lesion time, the lesion genesis time, the time at which it took to generate a lesion in the experiments most strongly was explained by animal volume and aquarium flow rate, all right? And most active or predatory species died faster than less active species. And we also found some evidence from genetic studies to show that high affinity cytochromes, cytochrome P450, 2J6, which is basically a type of cytochrome which is involved in scavenging oxygen. It was really, really high in the diseased animals. It was absent in the healthy animals. And we also saw that in another study put out by a different group. And so there's all this sort of evidence that hypoxia was involved with this disease. But we also looked at their tissues themselves. And what we found is that we did what's known as stable isotope analysis of them. This is delta 15N. So basically under anaerobic conditions, 15N, there's two isotopes of nitrogen, there's 15N and 14N. Microbial processes select for the 14N, and so they enrich pools of 15N. And so under anaerobic conditions, get these huge 15N signatures. We found across most species, in fact, there was an enrichment of 15N in their tissues from 2013 relative to healthy animals. So the wasting ones were much higher than the asymptomatic ones, except for this one species, Leptosteres hexactus. We're still trying to work out exactly why that was. And then when we look to see, whether this is something that occurs on an annual basis, we wanted to sort of, the source of the organic carbon we hypothesized was in fact phytoplankton. There's a citizen scientist who goes diving every single week at a single site on Whidbey Island, Jan Kosian, and I hope he's here tonight. And basically, we use observations that he made and compared them to basically data that was collected by the Penn Cove shell fishery. And what became clear from it is that the mean date of first wasting that occurred every year was almost at or just on the decline of this large phytoplankton bloom. All right. And when we compared the amount of chlorophyll, which is a measurement of phytoplankton abundance, as well as dissolved oxygen, what we found is that mass mortality had a very much larger chlorophyll A in the 3 months before compared to those months that were not wasting. And also oxygen was significantly lower than in the pre-wasting. So we got lots and lots of evidence that this is being driven by phytoplankton blooms on the West Coast. All right. What's also interesting, we looked at temperature and precipitation. Precipitation, typically many of the sites were in a drought condition in 2013, leading up to wasting. And essentially that drought was broken. So what happens is you also, because of the drought, there were these unprecedented phytoplankton blooms in the Salish Sea and Southern California, and essentially higher temperatures as well. And so it's probable that you had some flush of nutrients out from terrestrial environments, which actually caused this bloom of bacteria on the surfaces of these animals. But what about temperature? So why do we see the temperature impacts? Well, seawater loses oxygen as it warms. Obviously, the warmer the seawater, the less oxygen it can carry. And basically, the temperature swings we saw in Santa Cruz led to about a 3 milligram per liter swing in oxygen conditions. And so the idea here is that if you have any sort of perturbation of ambient oxygen conditions because of temperature, that might exacerbate this condition. Also important to note, the bacterial respiration is enhanced with higher temperature. All right, so here's a figure that was put out by Jude Apple in 2005. This is basically the production rate or the doubling rate of bacteria. And then here's the temperature. And it's almost sort of a linear increase over time. So higher temperatures will substantially lower available O2 on the surfaces of asteroids through a combination of there being lower oxygen in the seawater in general, as well as a greater respiration rate. So the implication is that this will get worse under future climate scenarios. There's a couple of other things that I'm going to just skip through here, but I'll just mention that under hypoxic conditions, essentially these animals excrete a huge amount of ammonium, which is toxic to all animals in the marine environment. And usually it's removed by ammonia oxidation, which is an oxic process. Under hypoxic conditions, the bacteria that are responsible for this process can't do their job. And essentially you get this buildup of ammonium. And also there's another impact of sulfur as well. All right, so sulfur is sulfides are toxic. They build up under hypoxic conditions and basically result in toxicity. So it's not just the oxygen, it could also be this, 1-2 punch of toxins accumulating as well as low oxygen conditions. All right, so I just want to conclude with a couple of points here. How long has sea star wasting been around for? All right, we assumed in 2013 that this was sort of a new phenomenon and that never been seen before, and it was absolutely everywhere, and it was a totally novel thing. But in fact, starfish wasting has occurred for over 120 years. I mean, this was actually from the New Hampshire, I believe, or Rhode Island, I can't remember, the state of Rhode Island, Providence plantations. And essentially, it is a report that said that there was starfish wasting happening in 1898. I have noticed on certain lots of starfish dredged from different localities what appears to be a disease attacking the skin first and not infrequently eating its way through the body. Sound familiar? This disease attacks both the common star and the purple star. Some of the starfish in my cars at Kickamet River were so seriously affected that some of their arms were entirely eaten through and the ends dropped off. All right, so it has been observed for a really, really long time. Here's some more reports of it. This is a starfish in the Kattegat in Denmark in 1970. Asterias forbesii in 1939. I came across a report there as well. And then in the Mediterranean in 2008. So also, if you look in the contemporary day, big wasting that's happening in the Yellow Sea, more or less the same time as what had happened in the Pacific Northwest. Also Port Phillip Bay in Australia, Antarctica, New Zealand. And so it occurs widely around the world. So, it's a common phenomenon, right? This is a timeline, 1898 to present day, lots and lots of reports of wasting, including some total extinction of individuals. This Heliaster kubnichii was one of the most dominant starfish in Southern California. It is gone today as of 1979. All right, so where did we go wrong? So we know that it's a commonly occurring thing. It's occurred worldwide. It looks like, it's this result of hypoxia. Why did we think that it was a densovirus to begin with? And it turns out that there's so many different closely related densoviruses that are picked up by our molecular tools that it's probable that we're picking up on multiple different densoviruses. And this sort of proliferation of viruses as animals die probably made us think that that was actually what was the one virus that was responsible for it. So important to note that some viruses hijack hypoxia-inducible factors to proliferate. So basically hypoxia causes their new virus particles to generate. And control material in those experiments where we basically challenge them with wasting tissue modulates are absolutely loaded with dissolved organic carbon. And so the results in those challenge trials may actually be a result of nutrient enrichment. There's just no way to say. Moreover, the apparent transmissibility into aquaria are density dependence in nature. So you saw one die and they all died. It might just be a snowball effect of one animal dying, their organic carbon becoming available to the bacteria that are living on other starfish, and so on and so forth, all right? So it seems that this was sort of a misleading result. So lessons learned. Look at environmental conditions, not always obvious, and sometimes they are at the absolute micron scale. And it's important to collaborate with biological or chemical oceanographers when dealing with marine disease events. Focus on the general physiology of bacteria, the microbiomes. All right, lots talked about trait-based sort of microbiology. You can say that this is a distinct pathogen, this is not. There are many examples of genera for which there might be one pathogenic representative, but none of the other ones are. Okay, so you can't simply take the name of a bacterium and say that's its trait, at least for most groups of bacteria. And also don't assume that every disease is transmissible, even if it looks that way, isolated, physical, chemical, and microbiological factors that might cause heart attacks. All right, so future work, we are looking at different diseases, all right? So sponges have this weird unexplained disease whereby they start to die. And in fact, it occurs under nutrient-rich conditions. Might be that. Stony corals, this is black band disease. This has already been established to be associated with hypoxia. There's currently a disease occurring in corals in the Caribbean, which seems to be related to localized productivity as well, perhaps. And also holothurians. So this is a sea cucumber. About the same time as sea star wasting, they started to have this weird ulceration disease, which again, might be the same phenomenon. And so, is sea star wasting a disease, a condition, disorder, a syndrome? Sea star wasting is likely a manifestation of boundary layer hypoxia driven by locally enhanced bacterial activity, probably as a consequence of increased organic matter from both phytoplankton as well as decaying animals. The effect will rapidly snowball when populations are dense because of lability of the decomposition products. In other words, they're really easy for bacteria to eat. And it will get worse under climate change, all right? As sea surface temperatures increase, lower oxygen conditions, faster bacterial metabolism. So sea star wasting is a disease, but is not likely pathogenic or transmissible. So it puts into the category of something like arthritis. Arthritis is a disease, but it's not transmissible, and it's definitely not caused by a pathogen. And so this is just a figure, right? This is on the road in Dutch Harbor, Alaska. There's a sea star that's been run over. Is the car a pathogen, all right? So think about where conditions might lead to disease. And with that, I'd just like to say thank you to a few of the people in my lab. Callie, Elliott, Sid Lally Aquino was an undergrad working in UC Santa Cruz. Jordan is a new grad student. Ryan did a ton of work on this as well. He's at University of North Carolina, Wilmington. Technician Chris Torito, and a few others, as well as my funding sources. And with that, I would be happy to take any questions.
Wow, I feel like I should have been eating dinner. That was like murder mystery theater. That was such a great story. Thank you for that. So there are some questions. Did any of the sea stars recover after the wasting started? And I'm adding to that, like, what are mortality rates once it gets it?
So, so I can answer the second part first. So the mortality rate, most animals that were observed in field studies, when they started to show some sort of lesion, it was pretty much guaranteed that they were going to die. And there are a few species, though, that they sort of showed lesions and then they kind of recovered. So, for example, dermasterias, there are reports of some dermasterias out there that had some wasting signs, but they actually recovered. They seemed to be okay after a while. And some of the ones in aquariums, for example, they were okay. But for most of the sort of gelatinous ones, like that sunflower star that I pointed out, they're very, very gelatinous. There's not a lot of sort of bony structure in them. Once they had a lesion, they were totally gone. And sorry, the first part of the question was, I'm trying to remember.
Well, just if any of them recovered.
Oh yeah, okay. So starting in about 2014, 2015, we started to see a huge recruitment of juvenile starfish on the West Coast. And that was sort of encouraging to a point. We thought that perhaps there was some selection that happened, and there has genuinely been sort of a recovery of some of them, and they are genetically distinct from the ones that were there from before. So it suggests that there was some adaptation to whatever condition they're subject to. But unfortunately, a lot of those animals have disappeared. It looks like there was a wave and then they just kind of died off. And that speaks to the fact that we don't actively sort of see many of the tiny little starfish that are out there because they're hard to see in surveys.
Related to that question is there a way to cure it? Or is this natural and you don't want to cure it?
Well, two parts to that question. The first is, say you're in an aquarium and you have wasting starfish, they've got some disease signs and suddenly they're all just snowballing and dying. Prior to sort of our thinking right now that it's hypoxia, the treatment with antibiotics sort of worked for a while. You could actually treat them with broad scale antibiotics and it would keep them happy. But then as soon as you took those away, the animals would die. Now we've had a couple of reports of success which treat directly the hypoxia issue. One of them is essentially to basically crank up the aeration in those aquariums so that there's tons of oxygen. It's totally super saturated. And so they're probably able to breathe under that condition. The other is to treat them with very specific antibiotics which target those types of bacteria that we saw were really increasing in abundance over time. And so they've been deployed successfully in at least a couple of aquariums, and I haven't heard any mortality from those places since, and it's been quite a while. In nature, unfortunately, no. It's impractical pretty much to treat things on a broad scale with antibiotics. And as far as sort of organic matter loading, there's no practical way of getting rid of it. It's just a natural part of the marine environment. However, I am aware that there are currently efforts to try and select the captive breeding program, selectively breed them for resistance to wasting in general. They're ongoing at the Friday Harbor Marine Lab. But it's just, there will be selection out there. A resistant population will come through, better athletes, if you will, perhaps, and certainly the genetic data is lining up with the hypoxia thing. But yeah, as far as treating it, not much we can do.
Could the amount of shore surface runoff, such as increased nitrates, be an issue?
It could indeed. So one of the things about nitrate and other eutrophication that happens from human sources and just from runoff from agriculture, et cetera, is that it leads to phytoplankton blooms. And as a consequence of that, you probably got this increased organic matter loading on the surface of starfish. What's really interesting is if you look on the West Coast, there are a couple places, Cape Perpetua springs to mind, where essentially they have the highest primary production on record. And they didn't see wasting. They all of a sudden saw wasting back in 2014, even in this presence of this huge amount of phytoplankton. So there's still a bit to work out as to how animals are adapted to their environment, their productivity environment. And that'll be interesting to focus on in the future.
Have you done aquarium experiments where you use an O2 atmosphere in excess of 0.21 atm and thus raise the dissolved oxygen level in the seawater and thereby reverse wasting?
So that actually speaks to the what I was recommending to veterinarians and animal care people at the public aquaria. We didn't actually, I don't believe that they mentioned, they measured the absolute oxygen concentration, but I told them to put in like 20 air stones into a tank and basically bubble at a very high rate. So I think that, and they've had success with that. So my feeling is that we haven't been able to do that in our studies as yet, but that would actually be an interesting thing to look at in the future if we can experimentally see what happens when we crank up the oxygen. It does have deleterious effects too when you have more oxygen in seawater as well. I mean, oxygen toxicity, the reactive oxygen species, oxidative damage all go up, but at least maintaining sort of an oxic condition is, or perhaps slightly more than saturated might work.
Jim Coyer asks, what is the deepest recorded instance of wasting, and does depth play a role?
Big question. So they have observed wasting on subtidal surveys with the trawl surveys, basically. So looking at Pycnopodia, for example, they did trawl surveys throughout the Salish Sea, and I believe that those were in 100 meters, 200 meters of water, and some in the not San Juan Islands, what's the Strait of Juan de Fuca. And they saw big declines there. They didn't actually observe wasting, but they saw the effects of wasting or some sort of population level change. And so it's probable that it occurs all the way down there as well. Most of what we know about sea star wasting comes from intertidal surveys, simply because we got citizen science groups involved in it, a few diving groups as well, but very few sort of systematic subtidal surveys of sea stars using, say, manta toes or diver toes.
All right, in the interest of time, we're going to stop there, but do not fear people who there's still 4 or so questions. We're going to send those to Ian and he will email you back. He has generously offered to do that. So don't worry, your questions are great, and Ian will get back to them. For all of you who have been as fascinated by this as me, this whole thing has been recorded, and it will be posted to the Shoals live stream page on our website, which is shoalsmarinelaboratory.org. So please feel free to share it. I found it really interesting. I bet a lot of people would find that interesting. So it'll be there. And on that same website, you'll find all of our events and other talks happening this summer. Our next talk is next Tuesday, the 23rd, same time. Brian Scarry, who is a National Geographic photographer, if you've seen a Nat Geo picture of a whale or a shark, it probably has Brian's name next to it. He is very charismatic, and he will be talking about sharks, which are also charismatic. So it's a very fun talk. It's probably the most popular talk at Shoals as far as the students are concerned, because the photos are amazing. So you can find out about all of these things on our website, shoalsmarinelaboratory.org. If you care to make a donation to support our work, you can also find information for doing so on that website. Thank you all so much. Thank you, Ian, a lot for that fabulous talk. Everyone have a great night. Take care, and we hope to see you again here soon. Good night.